Hi friends, today we shall be discussing about insulin resistance and the risk of type 2 diabetes mellitus, the risk factors and the development of type 2 and the role in the development of type 2 diabetes mellitus. Myself, Dr. Santosh Abraham, I'm working as a specialist registrar in endocrinology and diabetes at Scarborough General Hospital. We shall discuss today this topic under two major headings. I will go through the basics of insulin signaling once again, and I will discuss about the mechanism of insulin resistance in type 2 diabetes mellitus. Under insulin signaling, we will discuss about the effects of insulin binding after it binds to the insulin receptor, the AKT and the SHC pathways, and changes in insulin receptor after insulin binds to it, the insulin receptor substrate, a dual phosphorylation strategy for insulin receptor and the insulin receptor substrate, substrates other than the IRes, a short note on PI3K and AKT pathway, and downstream events after the AKT activation, a short note about FOXO factors, and a note on GRP2 SOS RAS MAPK pathway, and the importance of inhibitory serine phosphorylation. So, what are the effects when insulin binds to the insulin receptor? When insulin is activated, when the insulin and the IGF-1 receptors are activated by the ligands, they start a cascade of phosphorylation events. There is a conformational change and autophosphorylation of the receptors occur. This leads to recruitment and phosphorylation of receptor substrates such as IRes and SHC proteins. SHC actually activates the RAS MAPK pathway, which we shall discuss, whereas IRes proteins mostly activate the PI3 AKT pathway by recruiting and activating PI3K. So this is important. SHC is for the RAS MAPK pathway, while IRes is for the PI3K AKT pathway. And the PI3K activation leads to a generation of PIP3. Once you have the PIP3 formed, the membrane-bound PIP3 now recruits and activates PDK1, and this phosphorylates and activates AKT and atypical PKCs. So AKT mediates most of the insulin's metabolic activities, regulating glucose transport, lipid synthesis, gluconeogenesis, and glycogen synthesis. Also, it plays a role in the control of cell cycle and survival. Whereas the SHC, GRB2, SOS, RAS, RAF, MAPK pathway controls cellular proliferation and gene transcription. So whereas the SHC pathway is mainly concerned with proliferation and transcription of genes, AKT is involved in glucose transport, lipid synthesis, gluconeogenesis, glycogen synthesis, and also cell cycle control. So what happens when insulin is binding to the insulin receptor? Insulin binds to the alpha subunit, then the insulin receptor undergoes a conformational change. So it results in the activation of kinase of the beta subunit. And then it leads to transphosphorylation among the beta subunits. It further activates the kinase. It autophosphorylates the kinases and allows the recruitment of receptor substrates like IRes. What is insulin receptor substrate? So it is a signaling adapting protein, and through their function as protein, they function as protein scaffolds to organize signaling complexes. That is the importance of IRes. They are basically scaffolds over which the signals are organized. And these IRes transmit signals from insulin and IGF-1 receptors to make a cellular response. The IRes doesn't have any enzymatic activity of its own. A dual phosphorylation strategy of the IRes and the insulin receptors. Upon engagement of the insulin or IGF-1 receptors to their respective ligands, IRes 1 and 2 is rapidly phosphorylated on the tyrosine residues, but more slowly on serine or threonine residues. This is very important. It's rapidly phosphorylated on tyrosine, but it's more slowly phosphorylated on the serine threonine residues. And The serine threonine residue phosphorylation is done by a number of cytoplasmic kinases, which includes PKC, that is a protein kinase C, and JNK, and PPL70S6 protein kinase. What is the importance of the serine threonine phosphorylation? It provides a strong negative feedback signal as it is thought to block further tyrosine phosphorylation. So when the serine threonine phosphorylation occurs, it acts as feedback signal by blocking further tyrosine phosphorylation, and in some cases it also causes the degradation of the IRes itself. So it prevents overactive phosphorylation of the IRes, and thereby it has a negative exerts a negative feedback on the insulin signaling. So which are the proteins other than the IRes? Insulin and IGF-1 receptors can also phosphorylate other substrates. These are mainly the SHC proteins. When they are tyrosine phosphorylated by the IR, it results in activation of the RAS-ERK pathway. Then there are also GRB2 associated proteins, which recruits GAB, and GAB also are substrates for insulin receptors, and IGF-1 are. And finally, there is APS and CBL, and they recruit other proteins such as CAP to the insulin signaling complex. And CAP is important in the insulin stimulated glucose uptake, probably in the GLUT translocation. So again, when coming to the PI3 AKT pathway, the importance, this is a critical pathway which links the IRS proteins to insulin. So tyrosine residues are already phosphorylated. Then these residues bind to the SHT domains on the regulatory subunits. This further leads to the activation of catalytic subunit, and then it phosphorylates PIP2 to PIP3. Thus is PIP3 generated from the tyrosine activation of the IRS protein substrates. And PIP3 recruits AKT, and AKT is activated by phosphorylation, and again AKT is the final agent for inducing this downstream signaling. And once the AKT is activated, downstream events occur by mTOR C1 activation. So AKT phosphorylates TSC2, that is tuberous sclerosis complex protein 2, and then it activates the mTOR C1 complex. But mTOR C1 can also be activated via phosphorylation of PRAS40. So it is an inhibitor of mTOR C1, and when the PRAS40 is phosphorylated, the inhibition on the mTOR C1 is relieved. In other words, when the PRAS40 is in a dephosphorylated state, it acts as an inhibitor of mTOR C1. So when you phosphorylate PRAS40 by AKT, when AKT is activated, it relieves the inhibition. So by phosphorylating the TSC2, it activates mTOR C1, and by phosphorylating PRAS40, it relieves the inhibition on mTOR C1. So mTOR C1 is finally activated, and mTOR C1 phosphorylates and inhibits 4E binding protein, 4E-BP1, and it also activates ribosomal protein S6, kinases S6K1 and S6K2, and SRE-BP1. So these are all the effects of mTOR C1, that is a 4E-BP1, S6K1, S6K2 activation, and SRE-BP1 activation. And then this leads to a regulation of a group of genes which control the metabolism, protein synthesis, and cell growth. This is eventually what happens when the mTOR C1 gets activated. What are facts of factors? So they promote gluconeogenesis and negatively regulate the expression of genes that promote glucose utilization, including those involved in glycolysis. In other words, these are inhibitory normally. So these promote glucose utilization. These are inhibitory on the insulin signaling. So these promote glucose utilization, including those involved in glycolysis, HMP shunt, and lipogenesis. These promote glucose utilization by regulating the expression of genes. So more genes will be expressed, which will signal the glycolysis, which will be responsible for the production of enzymes involved in the glycolysis, HMP shunt, and lipogenesis. So when AKT phosphorylates FOXO, the FOXO is inhibited, and FOXO in the normal state tries to inhibit the IRS and the insulin receptor activation. So when the FOXO is phosphorylated by AKT, it inhibits this FOXO function. That is, FOXO no longer can influence its effect, and the inhibitory effect of FOXO on the insulin receptor and the insulin signaling is removed, and the FOXO is prevented from translocating from the cytoplasm into the nucleus. Another pathway is GRP2 SOS RAS MAPK pathway. So this is second essential branch for insulin IGF signaling. So it is activated independently of the PI3 AKT pathway. It has no relation with the PI3 AKT pathway. When the receptors are activated and the IRs proteins are activated, they possess docking sites for adapter proteins that contains SH2 domains such as GRB2 and SHC. So when the GRB2 are in turn activated, it will bind to GAB1 and SOS. And what is the importance of SOS? SOS activates RAS, which will ultimately result in the activation of MAPK pathway. And MAPK, once MAPK is activated, it causes increased cell proliferation or differentiation and regulates gene expression. So that is how insulin acts through this pathway. So ultimately, through this pathway, GRB2 to MAPK pathway, there is cell proliferation or differentiation. The genes regulating those expressions are actually expressed more. So you can see in this picture how insulin exerts its actions either through non-IRs or through IRs. So non-IRs is actually, it comes through the recruitment of CAP and CAP signal, and it goes for the, in turn, it causes more translocation of GLUT4 receptors to the cell membrane. And also, you can see that when IRS is recruited to the PI3 AKT pathway, it again results in the GLUT4 translocation. And at the same time, FOXO is inhibited, and the FOXO mediated degradation of the genes is actually inhibited here. Again, through the IRS, again there is the this is the GRB2 RAS MAPK signaling. So it results in the expression of these genes. And also there is an independent pathway. I mean, that is this is also mediated through IRS, but this is actually non-IRS mediated mainly. So this happens via the SRC proteins recruitment, and then the SRC gene recruitment, and then the GRB2 MAPK pathway, and it again results in control of cellular proliferation and growth here. So through the IRS and the non-IRS mechanisms, it results in GLUT4 translocation, and also in cellular proliferation, differentiation, and also in in the expression of genes controlling gluconeogenesis, lipid metabolism, gluconeogenesis, etc., glycogenolysis, etc. So this is in short another picture showing what happens when the insulin binds with alpha receptors. There is conformational change in the beta receptor. It gets phosphorylated. It also phosphorylates IRS. So IRS through the GRB2 SOS pathway, it can exert its effects on the cell cycle. And also another direct pathway is also shown here through the JNK. It influences the cell cycle. And independent of the IRS is another pathway here, which will through the GRF2, through the CAP GRF2, it results in the recruitment of translocation of GLUT4 vesicle and glucose uptake. Now the main pathway is through the mTOR, that is again coming through the IRS as well. So it will result in when the mTOR is activated, there is increased glucose uptake, and also there is glycogen synthesis. Okay, and in short, it inhibits gluconeogenesis, and it will cause the protein synthesis and growth. And you can also see the TSC2, TSC1, which again activates the AKT and then produces this effects here. Also, when this is activated, the FOXO is the FOXO, the inhibitory effect of FOXO, usually when it produces nucleogenesis, gluconeogenesis is removed and FOXO is sequestered. So gluconeogenesis will not happen here. So what are the negative regulators of insulin signaling? Intensity and duration of insulin and IGF-1 signaling plays an important role. So how these hormones will act will depend upon the intensity and duration of insulin and IGF-1 signaling. Signaling is attenuated by several phosphatases, and these dephosphorylate the receptors. So the phosphorylation of the receptors, especially the tyrosine receptors, which will lead to their activation, while dephosphorylation with phosphatases can lead to their inactivation. So it will in turn dephosphorylate the receptors. IRes proteins, PKCs, and the ERK or PI3 will their activity will be shut down. In addition, stress kinases such as JNK, IKK, and ERK, as well as PKCs or S6K, they also inhibit insulin IGF-1 signaling by inducing, as I've already told you before, inhibitory serine threonine phosphorylation. So the stress kinases, they inhibit insulin IGF-1 signaling by inducing the serine threonine phosphorylation of the insulin receptor and insulin growth factor receptor and the IRes proteins. Additionally, TRB3 inhibits AKT, and also it will inhibit SOCS and GRB binding to the receptors. And also GRB binds to receptors and IRes proteins, and thus inhibit signaling by competition. So TRB3 mainly inhibits AKT and SOCS and GRB, and it will eventually result in less receptor activation, and it will inhibits by competing with the AKT. So what are the phosphatases involved here in dephosphorylation? So there are two kinds of phosphatases: cytoplasmic protein tyrosine phosphatases and lipid phosphatases. These cytoplasmic phosphatases are mainly PTP1B, and also another transmembrane phosphatase called LAR. They dephosphorylate the tyrosine residues on activated insulin receptor and IGF-1 receptor as well as IRes proteins, and then they reduce their activity. Lipid phosphatases also regulate insulin signaling by modulating PI3K levels. So it has got a different mechanism. The PTEN will dephosphorylate the PI3K, and then it antagonizes the PI3K signaling in cells. So it decreases the activity of the PI3K. And SH2 domain containing inositol phosphatase, it's called SHP1 and 2. They also dephosphorylate by PI3K. So the lipid phosphatases, mainly the PTEN and the SHP, will dephosphorylate PI3K. What about the GRB10, GRB14, and SOCS and tribbles homologue, that is called the TRB3. GRB10 and GRB14 are adapter proteins. They are in the cytoplasm, and they decrease the insulin receptor and the IGF-1 receptor activity by preventing the substrates to bind with their activated receptors. That's how GRB10 and GRB14 work. They will prevent the substrate to bind with the activated receptors. So in short, the insulin receptor and the insulin receptor substrate cannot bind together. That's inhibited by the GRB10 and GRB14. SOCS protein, like SOCS1 and 3, will negatively regulate the insulin signaling. And tribbles homologue 3, TRB3, it's a false kinase, pseudokinase, and it also functions as an adaptive protein like the GRB10 and GRB14. So it disrupts insulin signaling by binding to AKT, and then it blocks its activation. So that's how tribbles homologue will work. An important concept is inhibitory serine and threonine phosphorylation. Tyrosine phosphorylation is essential for activation of insulin receptor and IGF-1 receptors and other IRs also. On the other hand, when you phosphorylate serine and threonine residues of the receptors or the IRS proteins, it will turn the insulin signal down or switch off the insulin signals. So increased inhibitory serine or threonine phosphorylation of the insulin receptors especially occurs, and especially of the insulin receptor substrate occurs when there are more cytokines, that is in the presence of inflammatory cytokines, then FAS, when there is decreased glucose, when there is hypoglycemia, and also when there is an insulin itself, and when in the presence of mitochondrial dysfunction and the presence of ER stress. So when there is less sugar, when there is insulin itself, and when there is mitochondrial dysfunction, and when there is ER stress, or when there is lot of cytokines released, inflammatory cytokines released, they will increase the serine threonine phosphorylation of the IR and insulin receptor and the insulin receptor substrate, and thus it will turn the insulin signaling down. So it is protective, because if there is more hypoglycemia, if you proceed with the insulin signaling, that means hypoglycemia will become worsen. And if there is more insulin also, if you proceed with the insulin signaling, that means more insulin will be binding the receptor. So in that way, it will produce a desirable feedback inhibition. But whereas the response from the inflammatory cytokines, mitochondrial dysfunction, and the ER stress, endoplasmic reticulum stress, is actually unwanted, when that can be detrimental to the cell when that happens, because it doesn't happen in the normal way, that inhibition. And serine phosphorylation, it definitely interferes with the function of the IR and the insulin receptor and the insulin receptor substrate. And when it's serine phosphorylated, these are targeted for inactivation or proteasomal degradation, as I've shown you before. And especially phosphorylation on 302 and 307 in IRs disrupt the function by inhibiting the interaction between IRs and PI3K. So the phosphorylation serine residues 302 and 307 in the IRs, that's very important. They inhibit the from IRS protein interacting with the PI3K, and thus they selectively inhibit the activation of the downstream signaling. And now let's come to the topic on insulin resistance. The pathogenesis of insulin resistance. What causes the insulin resistance, and what happens when there is insulin resistance. So first I will talk to you about insulin resistance in detail, in a small detail, and then I will talk to you about how to measure this insulin resistance. So it could be through this hyperinsulinemic euglycemic clamp, or there could be alternative approaches using the disposition index or the HOMA-IR and the QUICKI. Then what are the mechanisms of this insulin resistance? So already we know about the insulin signaling, so I will briefly tell about these mechanisms. We have already learned about how insulin signaling and how the inhibition of insulin signaling happens. And then what is the role of abdominal fat and insulin receptor resistance actually, and what is the effect of hyperinsulinemia on insulin resistance, and what happens when there is nutrient overload, like how it causes the insulin resistance, and why is insulin resistance more when the adipose tissue, especially when you have the visceral fat increasing, why it's more, and the reasons for this, like adipocytes are pro-inflammatory, so I will explain how it happens, and what happens when there is ectopic lipid accumulation, and how it contributes to the insulin resistance, and ectopic lipid accumulation, such as lipid accumulation, skeletal muscle, or in the liver. And finally, I will talk to you about the endoplasmic reticulum stress, the ER stress, and the unfolded protein response in that. So, and then I will talk to you about innate immunity and inflammasomes, mitochondrial abnormalities, I mean, these are all associated with the insulin resistance. So how these contribute to the insulin resistance and the role of ADP ATK, ADP ATK ADP ATP ratio, and how much the ADP ATP ratio will determine the insulin metabolism. And then I will talk to you about the skeletal muscle in insulin resistance, like what happens to skeletal muscle, the normal glucose metabolism according to Randall's hypothesis, and challenges to Randall hypothesis, and the skeletal muscle fatty acid metabolism in a small detail, and then how glucose influences skeletal muscle fatty metabolism, like hyperglycemia, how it influences skeletal muscle fatty acid metabolism, and finally a short note on gut microbiome and metabolome. So we all know that insulin resistance is the presence of an impaired biological response to either insulin administered exogenously or endogenously secreted. So this is manifested by decreased insulin stimulated glucose transport. So insulin usually stimulates glucose transport, so that will come down, and also there will be a decreased insulin stimulated metabolism also. Hence the metabolism in the skeletal muscle, because the glucose transport is less, the metabolism in the skeletal muscle also will be diminishing. And also there will be impaired suppression of adipocyte lipolysis. Usually insulin suppresses the adipocyte, the lipolysis in adipocyte. So when there is insulin resistance, insulin cannot do this function. And also insulin normally suppresses the hepatic glucose output, but when the insulin resistance happens, it cannot suppress that glucose output, and that's why you have a increased fasting glucose in diabetics, especially in type 2 diabetics. And the insulin resistance could in turn lead to multiple metabolic dysfunctions involving amino acids, their pathways, glucose metabolic pathways, and the lipid metabolic pathways. So how to measure this insulin resistance? The gold standard is called the hyperinsulinemic euglycemic clamp. So but it is very this technique is very cumbersome, and it's very complex. Actually, it's quite difficult, and it consumes a lot of time. But this is a gold standard. This was developed by Thr. So the patient is given a constant infusion of insulin, which will produce hyperinsulinemia. At the same time, glucose is infused to produce euglycemia. It's just like a sliding scale. And the glucose infusion rate represents the rate of glucose uptake or disposal into muscle, fat, and other tissues under hyperinsulinemic conditions. So how much glucose are you infusing in a given time? It will indicate how much glucose is taken by either muscle, fat, and other tissues when you produce the hyperinsulinemia. So thus you can measure the insulin sensitivity of the patient. But other approaches, because of this it is so cumbersome, other approaches have been developed. So another method is called the Reven steady state plasma glucose method, or the Beckman minimal model of glucose disposal. So it is actually done by a frequently sampled intravenous glucose tolerance test. So that's how you do it. So when you do the intravenous glucose level with the and then you measure the insulin level simultaneously, you can easily do the disposition index. So it is a combined measure of insulin sensitivity and insulin secretion. That is, in response to the glucose load, how much insulin is secreted and what is the insulin sensitivity can be measured. So it measures these two factors together. So that's why it's called the disposition index. So these are ECA methods, the Riemann or the Beckman models. And in research, you do the HOMA-IR and the QUICKI methods. So HOMA stands for homeostatic model assessment of insulin resistance, and it can be calculated using fasting glucose and insulin levels, and it is based on mathematical models, like there is reciprocal logarithmic calculators for HOMA-IR. So it correlates basically with the hyperinsulinemic euglycemic clamp. So it overcomes the difficulties there with the HOMA-IR. Other methods such as QUICKI, which is quantitative insulin sensitivity check index, is also a mathematical model. So it is also based similar to the HOMA-IR. As I've already told you before, you know the mechanism of insulin signaling, which will activate it and which will inhibit it. So the activities of serine threonine protein kinases are central. I mean, that's more important. So they catalyze this inhibitory phosphorylation of the insulin receptor or the IRes. So these become elevated, and enhanced dephosphorylation of the tyrosine residues by lipid phosphatases or cytosolic phosphatases can attenuate the insulin signaling. So I've already shown you before the PTP1B and the LAR will increase, and then it will dephosphorylate the tyrosine kinases. So these are cellular tyrosine phosphatases. Also, disruption of PTP1. If you disrupt it, it cannot dephosphorylate the tyrosine residue, and then you can see that the insulin sensitivity increases. That is, the insulin resistance decreases. What is the role of abdominal fat in insulin resistance? So abdominal fat and glucose tolerance is related, but it is independent of the total fat in the body. So abdominal fat is lipolytically very active than subcutaneous fat because it has got more adrenergic receptors, and hence it is it's lipolytically more active. And also the abdominal fat is more resistant to the anti-lipolytic effects of insulin. That is, insulin cannot exert its anti-lipolysis to a full extent in the adipose tissue. This is because of the alterations in the lipoprotein lipase activity. Thus, there is alterations in lipoprotein lipase activity, there is increased lipase activity, and hence there is a greater production of fatty acids which will be spilled into the circulation. And then high levels of HSD11 beta 1 in the intra-abdominal fat. This could result in the conversion of inactive cortisol to active cortisol, and then the cortisol will be also elevated. So this will produce changes in the adipocytes to increase lipolysis and alter the production of adipocaine. So when the cortisol levels are also increased due to increased visceral fat, it will increase the lipolysis, and also adipocaines are altered. But subcutaneous fat is associated with secretion of the adipocaine called adiponectin, which is actually beneficial in signaling. And what about hyperinsulinemia? So it causes insulin resistance. It is due to the increased concentration of insulin. First of all, it downregulates its own receptors, and also it desensitizes the post-receptor pathways. So these are the following mechanisms. As I've already told you before, serine phosphorylation of the insulin receptor and proteins inhibits insulin signaling. So there is increased ubiquitination and degradation of the IRes. And what about GRB10 and 14? They are negative regulators of insulin signaling. So mTOR will raise the GRB10 concentrations when the mTOR is excessively activated, when the receptors are activated. So it will raise the GRB10 concentration. And the GRB10 and 14 polymorphisms are associated with the development of type 2 diabetes mellitus when they are increasingly expressed. And ERK usually causes, that is extracellular signal related kinase, it will cause the feedback inhibition of insulin receptor. And also FOXO regulates transcription of insulin reception, and it inhibits the AKT action. So upregulation of FOXOs, if more FOXOs are expressed, then it will decrease the insulin signaling, and it will negatively regulate the insulin signaling. What happens when there is nutrient overload? It certainly causes insulin resistance. So cells will develop these. Cells will sense these nutrients with direct and indirect activation of transcription factors and protein kinases. So they usually sense the incoming nutrients like that. When they are sensing these, they get signals, and these signals provide inhibition on the insulin receptor through serine threonine kinase activation, So it includes the S6K, JNK, and the inhibition of kappa B kinase. So basically, when you have more nutrients, there is feedback inhibition of the insulin receptor through the serine threonine kinase activation. What about adipose tissue and insulin resistance? So when there is more excess nutrients, carbohydrates, proteins, or lipids are ultimately stored as triglyceride in the white adipose tissue. But if the storage capacity of this tissue is exceeded, lipids and other nutrients enter the non-storage tissues. So there will be ectopic lipid accumulation. This accumulation occurs in myocytes, that is muscle cells, liver cells, vascular cells, beta cells in the pancreas, and then they produce toxic lipid metabolites. These are the diacyl glycerol or ceramides. So this happens when the the storage capacity of adipose tissue is overwhelmed or exceeded, and when there is ectopic lipid accumulation. And the ectopic lipid accumulation will contribute to the insulin resistance. So I've told you about adipocytes. Again, they are pro-inflammatory. So they are more than storage cells. They uptake and release fatty acids. So it's a mobile pool actually, and they participate in the glycerol fatty acid cycle. They release leptin and cytokines that have lots of actions. So the adipocyte can be affected by accumulation of excessively nutrients. So it can be adversely affected. Adipocyte surface area will increase in obesity, and when the surface area increases, there is increased expression of leptin, IL-6, IL-8, MCP1 and GCSF. So these and other cytokines will attract macrophages actually, and then they release TNF alpha, and this produces this systemic inflammatory effect. So adipocytes, when when they are adversely affected by accumulation of excess nutrients, when there is overload in the adipocyte surface area will increase, and this leads to expression of the cytokines and leptin and interleukin 6 especially, and GCSF and IL-8, and these are pro-inflammatory. So these will attract the macrophages, and the macrophages come there and accumulate in these adipose tissues, and then they will start releasing TNF alpha, and then it will have local and systemic inflammatory effects, so such as the fibrosis in the liver. Those all could happen secondary to this. Coming back to the ectopic lipid accumulation, when as I told you before, when the lipid stores are when the storage of adipose tissue is overwhelmed, these lipids will accumulate in tissue such as muscle, liver, but these are basically not adapted to for storing lipids. So these are not actually mainly designed for lipids, for storing lipids. So these lipids accumulate here, and the excess lipids start to accumulate in these tissues which are not very well suited for storing lipids. So this is, as I have already told, this is called ectopic lipid accumulation. So when ectopic lipids accumulate, it will produce metabolic dysfunctions in tissues. So in muscle, insulin stimulated uptake is affected. So it is inversely related to the amount of intramuscular triglycerides. That is the intramyocellular fat. If it is increased more, then the insulin stimulated uptake will decrease, because there is already more energy representation in the muscle cells as triglycerides. So the cell gets a signal that no more glucose is needed. So thus the insulin insulin mediated glucose uptake will decrease, and hence in the muscle the insulin resistance will increase. That is, insulin cannot act on. So during the resting conditions, when in the well-fed stage, about 30% of the fatty acids from the plasma pool goes to the oxidation. They get oxidized, and the remaining 70% of the fatty acids form triglycerides. And when you have alterations in this balance between the formation and the oxidation, the formation of triglyceride and the oxidation of the fatty acid, this will lead to increased accumulation of triglycerides in the muscle cells, and increased lipolysis is associated with obesity, that will again increase the amount of fatty acids present to the muscle. So not only from the nutrient load, but also obesity itself, because of the increased surface area, there is increased adipokines and cytokines, and also there is increased lipolysis, because insulin, as I've told you before, insulin cannot act on this, the sensitivity of insulin to or the capacity of insulin to suppress the lipolysis in visceral adipose tissue is limited, or it decreases in obesity. So what happens is that there is increased lipolysis from this obesity, and along with that, if the individual takes a lot of fat, when the fat intake is increased, it will alter this balance. And when there is increased lipolysis from the obese adipose tissue, there is increased fatty acids, and then this will activate PKC isoform, and thus it will negatively feedback the insulin signaling. What happens when the lipid accumulates in liver? The simple answer is that you have the NAFLD. So when there is enhanced fatty acid delivery from adipose tissue by lipolysis, and also increased nutrient delivery by increased postprandial glucose concentrations, insulin resistance, the insulin resistance will increase. So these increased postprandial concentrations will provide this, and the increased fatty acids will provide excess substrates for the liver, and hence there is more acetyl coenzyme A and toxic lipid intermediates, such as I've told you, the ceramides and the diacylglycerol. So acetyl coenzyme A increases, toxic lipid intermediates increases. As you all know, if the acetyl coenzyme A accumulation increases, it will in turn again increase the synthesis of triacylglycerol. So lipid accumulation thus will in the liver is commonly seen in insulin resistance, and it will go it's usually called the non-alcoholic fatty liver disease. Another important aspect is endoplasmic reticulum stress, that is the ER stress, and consequently the unfolded protein response, all called the UPR. So the main functions of ER are post-translational processing of protein. So it will include the way how it folds the protein, because folding of the protein is very important for its function, and also it will affect the the ER also helps in maturation of the protein, processing of the protein, and there are also other quality checks such as quality control, and also it will influence the trafficking of these proteins to other cellular compartments. So there is in the protein transport, protein folding, maturation, and the processing and the quality of the protein, the ER has got an important function. It's all by the post-translational processing. So when the ER has got what happens is that when there is increased lipid accumulation, the ER usually accumulates excess levels of unfolded or misfolded proteins, and also it can happen stress also, like whatever stress you are having, like whether be it a physical stress or a cellular level of stress, it will accumulate excess levels of unfolded or misfolded proteins, or when there is increased production of these proteins, when there is need for that. So there is a distinct series of reactions occurring which will slow the overall protein synthesis, but the heat stress proteins or the chaperones will increase, and proteins also that increase the fidelity of the protein process, their synthesis is increased. Actually, to scavenge this unfolded or misfolded proteins. But eventually what happens is that even these HSPs or chaperones and the fidelity proteins which will help to remove these unfolded or misfolded proteins, their capacity also get overwhelmed. So the unfolded proteins will start to spill off from the ER. So there will be an unfolded protein response eventually. And there are three pathways for this unfolded protein response, and they're actually trying to alleviate the ER workload, because because these proteins are not actually very effective, so they want to decrease this ER workload because ER is working more here. So there is a RNA dependent protease kinase called the PERK pathway. There is a IRE1 pathway, and there is activation of transcription 6. So these three pathways actually mediate the unfolded protein response. So the unfolded protein response is basically to decrease the ER workload actually, because when there are more proteins needed, when there is more demand, these proteins are not mainly functional. So what happens is that the unfolded protein response will occur, and this is actually through three of these three pathways. And what happens when there is unfolded protein response? So it will result in the activation of JNK and the nuclear factor NF kappa B in activation. So this will result in the decreased IRS1 activity. So when there is more unfolded protein, there is unfolded protein response in order to alleviate the ER workload. And but when there is a secondary to this response, there is activation of the JNK and the NF kappa beta and NF kappa B, and then which will result in the decreased IRS1 activity. Also, the unfolded protein response will result in increased levels of inflammatory mediators. So it is through transcription, alteration in transcriptions, and also it will reduce hepatic gluconeogenesis. And after prolonged activation, it finally will cause the cellular dysfunction and apoptosis, because these unfolded proteins are not they are not functional, and the ER is producing a lot of unfolded proteins. So the cell sense that actually the cell is basically having a problem here, because it is not working in the normal fashion where there should be more functional proteins. So when the cells sense that, they automatically undergo the apoptosis. So but that is through this unfolded protein response. So basically they will decrease the IRS activity, they will increase the inflammatory mediators, and also they will decrease the hepatic gluconeogenesis, and ultimately they will cause the cellular dysfunction apoptosis. So this is associated with increased this is actually associated with the insulin resistance actually. And innate immunity. So when there is activation of the immune system in infection, obesity, it shows insignificant insulin resistance. So you know that when there is acute infection, there could be a compensatory hyperglycemia happening. It's not a compensatory hyperglycemia actually. It could be there will be a secondary hyperglycemia actually happening because the pro-inflammatory state can actually increase the insulin resistance, just like the obesity. So so especially in obese individuals, when they are sick, you could see that the glucose tolerance is impaired. So this is actually mediated by elevation of the fatty acid levels. So toll-like receptors, especially TLR2 and TLR4, they normally respond to the bacterial cell lipids to induce the innate inflammatory response. They are also activated by these fatty acids. So basically the toll-like receptors, they produce the inflammatory response in response to bacteria, but when the circulating fatty acids are increased, like in obesity, they are also activated. But another important point is that PUFAs, like the polyunsaturated fatty acids, will turn down the TLR signaling. So when the TLRs are again activated, as in infection or obesity, it will result in the insulin resistance. And another important concept is inflammasomes. So when the innate immunity is activated, there is increased production of inflammasomes. So these are large multi-subunit protein complexes, and they are under the control of caspase 1 usually, and sorry, they control the release of caspase 1, and also they are important in the maturation and secretion of interleukins, especially IL-1 beta 1, and they have potent pro-inflammatory actions. So these are a risk factor for development of T2DM. Like when the inflammasomes they are more expressed, or they are more produced, it will increase insulin resistance, and further it will increase the risk for type 2 diabetes mellitus. So they actually do by disrupting the beta cell function, the inflammasomes. That is, they will directly go and disrupt or block the beta cell function. And then there are mitochondrial abnormalities. So when you examine the mitochondria, in the ratio of mitochondria in the muscle fibers, oxidative type 1 fibers to glycolytic type 2 fibers is decreased. Actually, the oxidative fibers are required because they produce more energy when compared with the glycolytic fibers. So there is 30% decrease in the mitochondrial oxidative capacity, and also there is increased almost about 80% increase in the intramyocellular lipid compartment. So this happens. So this causes this mitochondrial abnormality when there is intramyocellular lipid content increase, and up to 80% it will increase, and it will result in almost 30% of the mitochondrial oxidative capacity. So that could result in decreased formation of oxidative fibers, type 1 fibers. And also there is decreased expression of nuclear encoded genes which regulate the formation of mitochondria, such as PPAR coactivators such as 1 alpha and 1 beta. So they are called PGC1 alpha and PGC1 beta. Again, the activity of the electron transport chain, that is also reduced, and studies have shown that intramyofibrillar mitochondria, that is the mitochondria in the fibrils of the muscles in type 2 diabetic patients, are smaller than the normal. So when the mitochondrial activity, the oxidation of mitochondrial, the oxidative capacity of the mitochondria is compromised by increased lipids and by the decreased expression of certain genes which produce the mitochondria, mitochondrial mass will decrease, and also there is the mitochondrial oxidative capacity will also be decreased by the decreased suppression of electron transport chain also. So these all are associated with insulin resistance and type 2 diabetes mellitus. And when there is in sedentary individuals, there is less energy demand, so there is a decrease in ADP ATP ratio. So this actually will impair again the electron transport in mitochondria. So increased levels of reduced form of NADH, that is NAD, that is the NADH, also will impair the TCA cycle because there is no much oxidation. So in NADH accumulates, so that will again impair the TCA cycle. So this will result in the generation of reactive oxygen species, and that will further compromise the mitochondria. Another important aspect is acyl carnitine and the post-translational modification. So when there is impaired mitochondrial oxidation, the acyl carnitine concentration also will increase. When the acyl carnitine concentration increases, they represent actually the markers of insulin resistance. So when the acyl carnitines increase, they usually indicate there is impaired mitochondrial oxidation because the carnitine is not functioning well actually. So that's why the acyl carnitines, when you cannot transfer the fatty acids from the cytosol to the mitochondria, their concentration is increasing here. So it will serve as a marker of this resistance, insulin resistance. And also the mitochondrial proteins, when they are when their post-translational modification is affected by decreased activity, such as when they are acetylated by decreased mitochondrial enzyme activity. there is increased intramitochondrial NADP. So one of the proteins is SIRT3. It primarily deacetylizes, it removes the acetyl groups of the mitochondrial enzymes, and thus its activity increases in the exercise, when there is increased use of ATP, and when there is increased production of oxidized NAD. So when this acetylation is decreasing, there is decreased mitochondrial activity. There is more reduced NADH in the mitochondria, which will add more stress to the mitochondria, and which will suppress the electron transport chain, in fact. So when the proteins are modified by the mitochondrial proteins are modified by post-translational modification in when there is excess nutrients, especially by acetylation, it implies that there will be acetylation of the mitochondrial enzyme. There is a decreased mitochondrial enzyme activity. So the removal of these acetyl groups actually does not occur. So this is actually shown by the decreased SIRT3 actually, because in exercising muscles, the SIRT3 concentration increases, and it helps to remove this acetylated proteins, and thus it activates mitochondrial enzymes. So coming to the skeletal muscle insulin resistance. So when there is when we absorb the glucose, it is actually disposed in the skeletal muscle, and the primary the first mechanism in the glucose storage is formation of glycogen. In obesity, when there is insulin resistance, the insulin resistance in the muscle is manifested before abnormalities in the signaling of insulin in adipose tissue and liver. So you can why this happens is because there is limited nutrient storage capacity of the skeletal muscle. So the insulin resistance in the skeletal muscle is manifested. So that is the importance of skeletal muscle and insulin resistance. So it could be indicating the early, the early formation and the early development of type 2 diabetes mellitus, or it could be even giving us a clue that this individual is going to have the type 2 diabetes mellitus. So when you talk about this progression of impaired glucose tolerance to diabetes, we have called a Randall's hypothesis, where it is hypothesized that elevated fatty acids, they actually predict the progression from IGT to diabetes. So when there is increased fatty acids, there could be more risk of forming diabetes. So how they proposed this was like, when there is increased fatty acid to skeletal muscle due to increased lipolysis, it will result in decreased muscle glucose uptake. We all know that. So they told that these FAs compete with glucose for substrate oxidation in muscle. So increased FA metabolism will increase the ratio of mitochondrial acetyl coenzyme A to the cytosolic coenzyme A, and will decrease the NADH to NAD plus ratios. So it will when the NADH is getting decreased, it will inhibit the pyruvate dehydrogenase complex, and hence there will be it will lead to increased mitochondrial citrate actually. So the citrate will cause inhibition of phosphofructokinase. That is an enzyme in glycolysis, and it's a key enzyme. So the FAs actually, what the FAs do is that they compete for the glucose for substrate oxidation. So the FAs will increase the acetyl coenzyme A in mitochondria, and also it will increase the NADH in mitochondria. This will inhibit the pyruvate dehydrogenase complex, and hence the citrate will increase, and the citrate will inhibit the phosphofructokinase, thereby halting the glycolysis. So also increased G6P glucose phosphate inhibits hexokinase 2 activity, and it will increase. It will result in an increase in intracellular glucose concentrations and decreased glucose uptake. So what are the challenges to this hypothesis? So, but recent studies have shown that the primary effects of fatty acids when insulin level increase is a decrease in the glucose is a decrease in glucose transport because the intracellular glucose and glycogen is decreased. Here in Randall's hypothesis, they title that because of the inhibition of glycolysis and because of the increased glucose 6 phosphate, because the glycolysis is inhibited, there is an increase in intracellular glucose concentration. But the recent studies show that even though the increased insulin levels, even though in the insulin resistance there is increased insulin levels and there is decreased glucose transport, the intracellular glucose and glycogen is decreased. This is because it is due to the decreased activity of the PI3K and the increased activity of the PKC. So PKC will actually phosphorylate the serine residues, and then it will cause the degradation of the IRes units, and also there is unregulated translocation of NF kappa B into the nucleus. So these are all associated with the IR. So insulin resistance. So so what happens is that under increased insulin levels, because of the increased F phase, there is increased intracellular glucose, in contrast to the Randall's hypothesis, which told that because of the glucose 6 phosphate accumulation there was increased intracellular glucose. Even it has been shown that if you give aspirin or salstrates, it will inhibit this IKK beta pathway, and the insulin sensitivity is improved. Now let's come to the fatty acid metabolism in skeletal muscle. The uptake of fatty acids is mediated by 3 families of proteins: fatty acid translocase, fatty acid binding proteins, and fatty acid transport proteins. So there's a translocase, there's a binding protein, and there's a transport protein. So FABPs bind fatty acids, eicosanoids, and retinoids. So they facilitate uptake of fatty acids and promote intracellular transport to subcellular organelles. So they will regulate how the fatty acids will move from inside the cell to other organelles. So if the FABP levels, the binding protein levels decreases, free fatty acids will increase and the insulin resistance also will increase. Another checkpoint is CPT1, which is mainly involved in the fatty acid beta oxidation. So you know that malonyl coenzyme will inhibit the CPT1 in liver and decrease the beta oxidation of fatty acids. So in insulin resistance there is overexpression of CPT1 in muscle. Overexpression of CPT1. There is underexpression of CPT1 in muscle. So that will lead to decreased fatty acid oxidation. Even if you manipulate the CPT1 and if you causes overexpression of CPT1, it will cause increased fatty acid oxidation and it will decrease the triglycerides inside the muscles. And also the CPT1 mRNA is regulated by PPAR alpha activators, fat feeding, and also exercise. So and it is inversely correlated with the obesity. So all these things, the PPAR alpha activators, exercise, will increase the muscle CPT1 and hence it will increase the beta oxidation of fatty acids, whereas CPT1 expression will be decreased in obesity, and thus you can see that impaired fatty acid oxidation occurs, and this is now getting correlated with the insulin resistance. So what happens when there is hyperglycemia in skeletal muscles? Like what happens to the fatty acid metabolism? When there is increased uptake of glucose, when there is increased acetyl coenzyme, because the glycolysis will proceed forward, and there is increased tricarboxylic acid cycle activity, and so the TCA will proceed again, and there is oxidation of triglycerides and glucose, and finally there is increased citrate. So it will go back to the cytoplasm. This citrate will go back to the cytoplasm. It will activate acetyl coenzyme carboxylase, and it will increase the susceptibility of ACC to phosphatases. So increased acetyl coenzyme carboxylase will cause conversion of acetyl coenzyme to malonyl coenzyme. So you should remember that when there is increased glucose uptake, there is increased production of acetyl coenzyme, and hence there is increased production of citrate. and the citrate will come to the cytoplasm. It will activate ACC or acetyl coenzyme carboxylase, and acetyl coenzyme carboxylase will, since its activity increases, it starts converting more acetyl coenzyme to malonyl coenzyme. And what happens when there is more malonyl coenzyme? Malonyl coenzyme will inhibit the CPT1 or the CAT1 on the outer mitochondrial membrane. So when this happens, there is increased buildup of long chain fatty acids, acyl coenzymes and diacyl, and there is increased buildup of this long chain fatty acids on this side, and the diacyl glycerol also. So this will increase the fatty acid concentration in the blood, as well as it will activate also the PKC isoforms, and thus contribute to the increased insulin resistance. So this is actually happening when there is increased glucose. Once again, when there is increased glucose, there is increased acetyl coenzyme production, and this will result in increased citrate production also because of the increased TCA activity. And the increased citrate will again activate the acetyl coenzyme carboxylase, and already the acetyl coenzyme is in plenty. That will the increased acetyl coenzyme carboxylase ACC will convert the excess acetyl coenzyme to malonyl coenzyme, and the malonyl coenzyme will inhibit the CPT1. It will lead to the buildup of the long chain fatty acid coenzyme, and this will actually activate PKC isoforms, and that leads to insulin resistance. And finally, an important but a topic under research for what is the role of the gut microbiome and metabolism. So in the gut, the microbiota have major effects on intestinal barrier function. So they influence the breakdown of otherwise indigestible dietary components, and there is modification of also bile acids and other substances. Even there is development of the gut, and also processing of the immune system. So these effects can lead to release of bacterial proteins, endotoxins, cytokines in the bloodstream, as well as produce changes in hundreds of metabolic products, including bile acids. Like when these gut microbiomes are altered, it could result in release of, it is proposed that it could result in the release of bacterial proteins, endotoxins and cytokines. So when the gut microbiota are inhibited by antibiotics, oral antibiotics, it could result in the release of these bacterial proteins and endotoxins and cytokines into the bloodstream, and there could be a lot of metabolic changes. It is proposed, there is no evidence. So these bile acids, short chain fatty acids, amino acids and many other class of molecules, their metabolism is changed. So these will lead to metabolic dysregulation and immune activation, and ultimately it will lead to insulin resistance and progression of diabetic pathogenesis. It's all proposed, or it is all hypothesized. There is no definite proof for this, even though there are several metabolites, especially 2-amino adipate, alpha-hydroxybutyrate and acetyl glycine. They are shown to correlate positively with insulin resistance. That is, they are directly related, proportional. Their concentration increases with the insulin resistance, and they are suggested as biomarkers of type 2 diabetes mellitus. Thank you for watching this presentation.